this is yours.
Okay, thank you. Okay, I can't compete and I won't try with this beautiful presentation. I'm just here to say how happy I am that Jean has agreed to come and open this new, beautiful, I hope, long-lasting set of distinguished guests. And several people came to me asking, what is sociogenomics? And I said, yeah, come, you'll hear all about it. And no wonder they didn't know, because Gene Robinson invented this word. It didn't exist before him. So what are we talking about? We're talking about a new field of research that has initiated from a totally unexpected angle of the genome sequencing field and area. So there are several rules that would say who is worthy of special appreciation, special scientific appreciation. That would always be a person who preceded everyone else in the field in foreseeing the implications, the possibilities, and the impact that would become if something would be developed. And I'm here to tell you that Jean saw all that long before anyone else. Well, you came to our lab in 89, right?
A little after, 94. Yeah.
94, okay. So here is this bee expert writing me, I think that molecular biology would have a big impact on my field. And that was still the time when people were arguing, what is all this about the Human Genome Project? It's just banking evidence. It's not real research. We've heard very distinguished people saying that. And here is someone who jumped several steps ahead and said, not only would it be relevant, I think it will be relevant for me, and I'm willing to take the leading position to lead others to make it happen, which had been a really big commitment. It would only fit someone who hardly sleeps ever, which is a good example here in this case, and who's open-minded and is capable of taking the responsibility. And we as researchers, we're not only responsible to what we do, but also to what our students do, our postdoctoral fellows do, what happens in our field. It is a grave responsibility indeed. And we feel responsible for their lives ever after. The students don't realize that often, but this is the case. So Menachem Ben Sasson was once lecturing about the concepts that make new research develop. He talked about Judaica-related studies, but I think it fits every field. He said there should be always an initiator of a new field or direction of research and an expander. If you want to look in Israeli standards, so Hershko was the initiator, Chekhanouver in many aspects had been the expanders. The problem is Jean is both. He both initiated it and he expands this field ever since he made that strategic decision. And I tried to convince him to stay here and failed. And I think science gained, because we couldn't have invested in what Jean is doing as much as the United States could. So now the research spans many countries, many researchers, much more ambitious directions of research. Jean comes to us from Urbana, Illinois, but his network of links is all around the world. It's always a pleasure and an honor to have you come here. I won't tell anyone what is sociogenomics. That's your job. Thank you very much.
Okay, I got all the technology set, and now I can tell you how honored and pleased I am to be here, to be the inaugural lecture. Mona, thank you very much for the kind words. What Mona didn't say was that she took a chance on me to host a then quite young assistant professor just getting tenure for this new endeavor, and I learned a great deal in Mona's lab. She was very generous with her time, with resources, and it was only possible in the lab of a brilliant molecular neurobiologist that this work could have taken hold. So I will always be grateful for that. Professor Hen, I'm really honored to be here. Let me tell everyone that your slides are better than mine, much prettier, but I'll do the best that I can. And I also want to give a special shout out to my sister and brother-in-law who have come from Modi'in to attend the lecture, maybe to have a nap, maybe to listen, but thank you very much. So I'm going to talk about sociogenomics, and to do so, let me set up the problem by giving you this illustration. We're at still a very interesting point in time in history where for some traits, human traits, we're very comfortable thinking about the role of genes, and for other traits, there's still a lot of concern. So I illustrate this in this slide here. For sports, for athletics, we have no problem thinking and talking freely about the idea that genes are involved in those particular traits. The image on the top left is a father and two sons. Some of you that know American football might recognize the Mannings, Archie Manning and his two sons, Peyton Manning and Eli Manning. And as I say, football dynasty, and we're all very comfortable about that. When we get into other traits like intelligence, personality traits, there's not a uniform perspective. There are people that embrace the idea that genes are involved in those traits, and there are others that either don't believe it or they're quite concerned about the implications. They're concerned about something called genetic determinism, the idea that genes run the show completely. And we've seen this movie before, and it didn't turn out very well multiple times. So we have a legacy of shame to live down in the field. Some of the key events, low points in history: eugenics, the Holocaust, Marxism, other social and political movements that have been based either on one extreme view of human nature or the other extreme view, either nature completely or nurture completely. So we have a situation where genes and the environment, nature and nurture, are thought to be very different entities, and we have this competition between perspectives. What genomics has done is given us the ability to see the picture more fully. So on the top, you see the old view that there are hereditary influences on the genome and through the genome on behavior. So at the top you see genes acting on elements of the brain such as RNA and proteins, and then affecting brain systems and then leading to variation in social behavior. The lower panel is the new panel. That's the panel that has been revealed by genomics, and it's that aspect that I highlight in naming this new subfield sociogenomics. The idea that there is a two-way interaction between genes and behavior. Social information gets under the skin and affects the brain, affects the genome inside the cells in the brain to cause changes in behavior. And it's not one perspective replacing another perspective, it's that there are both perspectives, that nature and nurture both act on the genome. And so this really encapsulates the idea of sociogenomics right here, using an evolutionary perspective, the ability to learn from multiple species, a comparative neurogenomic perspective that allows us to gain this knowledge. So what are some of the roots of sociogenomics? We don't invent subfields or ideas out of whole cloth. science proceeds many ways, building on the advances of others. So here are two advances that, two foundational pieces of biological information that were very important to me as I developed these ideas. First of all, sounds simple, but it's rather profound, and that is that social behavior is influenced by genes and therefore shaped by evolution. This was the idea of E.O. Wilson, Edward Wilson from Harvard, articulated in his brilliant but controversial, as I'll talk about later, book, Sociobiology. And so this came from the field of evolutionary biology. Secondly, an important insight came from also a relatively new field, the field of a merger of evolution and development known as evo-devo, and that is that there is profound conservation, evolutionary conservation of gene function. That is, if a gene is involved in giving rise to a trait in one organism, it may well give rise to a very similar trait in another organism, even if that organism is very distantly related. So let me give you an example of that because it helps illustrate the nature of the sociogenomics perspective. So it is well known that the eye of the human, which is an image-forming eye, is really different than the eye in insects, even though the insect eye also forms an image. But because the last common ancestor of those two lineages did not have an image-forming eye, this example has always been seen as a textbook example of an evolutionary process known as convergence. The idea that distantly related organisms solve a biological problem in evolution independently, but they often converge on the same solution. So, under such a model, one would not necessarily expect to have any conserved molecular machinery underneath that. And that's kind of where the field was, and it made it very difficult to compare across organisms. Because if you know a little bit about the phylogeny and know that a trait was not inherited from a common ancestor, like the eye, then you wouldn't expect it. Well, no one told PAC6. PAC6 is a transcription factor. That means it's a gene that regulates the activity of other genes. And it turns out that in a really important experiment in the then young field of evo-devo from the lab of Walter Gehring in Basel in Switzerland, you could take the PAC6 gene from a fruit fly and insert it in a mouse that was made to be blind. And if you put the fruit fly's PAC6 gene in the mouse, it would direct the development of a normal, not fly, mouse eye. Conversely, the mouse PAC6 into the fly, and you get a normal, into the blind fly, eyeless fly, and you would then get a normal gene, a normal eye in the fruit fly. So, tremendous conservation of gene function. This will be a theme that you'll hear a little bit later on. So these two were really profound roots of sociogenomics. Then getting a little closer to our own work, a very important piece of work that was done in my lab in collaboration with the lab of Marla Sokolowski at University of Toronto, the work performed by Yehuda Ben Shachar, who got his first degree at Tel Aviv University and his PhD in my lab, relates to the two papers that you see in front of you. Marla's lab discovered a gene that was called the foraging gene. Its precise details are not important, but it's in the title on the left. And she found that this gene is involved in variation in behavior in fruit flies, but variation at the level of DNA, that is, inherited differences. Those flies that inherited one flavor of the gene, one form of the gene, technically known as an allele, would forage at a higher level than those that inherited another version of that gene. Yehuda took these ideas from this work, and we started to think about transposing them from DNA to RNA. So we start with DNA, that's where information is encoded. RNA is where it's transcribed. It is an intermediary molecule that eventually gets translated to protein. Protein are what builds bodies, rejuvenate bodies, allows us to learn, allows bees to fly. Proteins do all the work, as I say in my genes and behavior class. Genes are boring. There's no such thing as smart genes or mean genes or anything like that. They encode information that gives rise to protein. The wonder of biology is how proteins work together to be able to build and sustain and revive life in all its dimensions. So we had this DNA polymorphism. We asked whether we could transpose that, if you will, into a different key. Instead of talking about DNA, talk about RNA. And now we're talking about gene activity. And so if one version of the gene that's inherited by fruit flies makes them fly more and forage more, we wondered whether if we turned up the activity of that gene at the RNA level, whether it would create foragers at a younger age. And it did. So we were able to translate this information from DNA to RNA, and that gave us the idea that genes are acting at different timescales, hereditary changes and also changes that occur in real time in the life of the organism, which of course would be sensitive to the environment. Before I get starting on the actual research, let me amplify that and make sure we're all on the same page. I know we have a very diverse group here. The Institute for Advanced Studies is aimed at bringing together people from different disciplines. So here are the levels of organization in the brain. So the levels of biological organization that relate to behavior, starting from DNA, moving up to RNA, to proteins. We covered that, right? There'll be a quiz, so remember that. And then we get into the more traditional areas of the brain, the neurons, the brain circuits, brain regions, and finally behavior. Most of what we'll be talking about has to do with the activity of genes. That's measured at the level of RNA abundance and how that relates to behavior. But just to plant the seeds for some ideas we'll be talking about towards the end, just look at how many different levels of biological organization exist between RNA and behavior. It's really a long and winding road. So let's keep that in mind. Okay, so the honeybee. Why the honeybee? Here are some reasons for using the honeybee to address the question of the relationship between genes and social behavior and encompass the ideas that we now refer to as sociogenomics. First of all, bees have been studied for a long time, as we heard in our opening remarks. They've been of interest to humanity for a long time, and for good reasons. Here are a couple of historical tidbits for you. Humans used honeybees for the first alcoholic beverage ever and the first sweetener. And so, for obvious reasons, they became really, really important. And so people spent a lot of time observing them and learning about them to be able to maximize their ability to get those two substances. So as a result, thanks to many, many generations, hundreds, thousands of years of beekeepers and bee watchers and natural historians, we know more about the natural behavior of honeybees than any other organism except for ourselves. Fruit flies can be manipulated better in the laboratory. Mona's mice can be manipulated better in the laboratory genetically, but we know more about the natural behavior of honeybees than any other organism except ourselves. So we know this. We know this is a canon of knowledge that we have inherited from these generations, and as a result, scientists have been able to develop ways of manipulating the behavior, which is very important. We also, thanks to social evolution, have a bonus, and that is honeybees exhibit extreme behavioral states. Honeybees and other social insects, and indeed all organisms that live in advanced societies, have a division of labor. Some individuals do one thing while others are doing something else. And often they will specialize on these activities for extended periods of time. This allows us to better track the relationship between gene expression and behavior, because gene expression unfolds on the order of minutes to hours to even days. So neural activity, the activity of neurons, is measured in milliseconds or seconds. The activity of genes is slower. And so having behavioral states that are expanded and that have a longer period, a longer duration, helps us better track. So these extreme behavioral states are a gift. The social environment of honeybees can be precisely controlled. This image here that you see on the right, a set of beehives, each of those tall stacks is a beehive. A family of bees lives inside of each of those stacks. And we know every individual in that. We don't have to study who's, like in a primate society, who's really related to whom and what are the social groupings. Very simple with bees. They're in one of these colonies and one of these stacks. They're part of a colony between 30 and 50,000 individuals. And then we can manipulate those colonies on a large scale, at a replicated scale. We can perform social engineering, if you will, and control under controlled conditions. Therefore, we can get large sample sizes, and in the studies I'll be talking about, we're able to get sample sizes of 10 to 20 individuals across multiple colonies. We always replicate the work across colonies because the colony is actually a unit of organization. It's not just an aggregation. It's a unit upon which evolution has acted. So we have all of these attributes, and now, thanks to some really great work by a graduate student, former graduate student in the lab, now postdoc, Tim Gernot, we have a barcoding system where we're able to track individuals in an automated way and get high-dimensional behavioral data. One of the world's leaders in this area is Ofer Feinerman, whose name you heard mentioned before at the Weizmann Institute, is doing the same for ants. So we can monitor behavior and then really get large data sets and precise behavioral monitoring. And I'll show you how we're beginning to use this system. So that's a little bit about the honeybee system in general. The last bit of introduction that I need to make is to introduce you to the two behavioral systems that I will be drawing upon for tonight's lecture. The first one is hive defense, and the second one is social foraging. Hive defense, I've laid out some of the key ingredients of this for you. In brief, the honeybee is a victim of her own success. Honeybees are very successful at collecting nectar from flowers and turning it into honey. Now I ask you, if you're Winnie the Pooh and you want a meal, you could have a choice between going from flower to flower and gathering 1 or 2 microliters of sugar syrup that's about 40% sugar, or you could take your big fat paw and knock over a beehive and get, say, 50 kilos of honey, which is 85% sugar. So if you had that choice, what would you prefer doing? Hopefully you made the right choice, and you realize then that honeybees are at risk. There are many predators, including ourselves. Humans have been historic predators, other large vertebrates, small invertebrates. They've been really prime targets because of the fact that they collect resources that are distributed in the environment. They concentrate them in one place. So honeybees have evolved sophisticated defense systems. These defense systems involve hereditary and social influences. So that is, there are some strains of bees that are more aggressive than others. I'll talk about one briefly later, the Africanized bees. So bees that evolve in places where there are more predation threats evolve to be more easily aroused and defend their hive more vigorously. There are social innovations. There's division of labor, as I said, even for defense. So there are groups of bees that specialize on defending the nest. 2 groups in particular that we'll be using experimentally in a few minutes. Guard bees, they are at the entrance. So here's an entrance to a beehive, human-made beehive, but you could find the same thing in natural nests. These are bees that patrol the entrance to the colony. They look like this. They have their forelegs are off the ground, their antennae are forward, and they act as the bouncers, like at a bar. They can tell who belongs and who doesn't belong. If they detect an intruder, and they can detect who belongs in their colony and who doesn't, not just wasps or ants or bears, but also bees from different colonies, they will release an alarm odor, a pheromone, that alerts the rest of the colony. But the first group of bees that are alerted are the soldiers. They fly out and seek to sting the intruder. So a very sophisticated system. And then secondly, social foraging, also very sophisticated, which, as I said, gave rise to the success of the foraging enterprise in a honeybee colony. And I'll just give you a couple of key aspects there. Again, division of labor. So there are scouts that go out and look for the new food sources. And then when they come back, when they find something, they come back and communicate this to other bees, the recruits that wait to be recruited to a food source. So I'll tell you a little secret. Not all bees are busy as bees. Some are kind of hanging around waiting to be told what to do. And so the scouts are very key. They communicate by means of an incredible evolutionary innovation, the dance language, which I'll tell you a little bit more about in a few minutes. And then they will then direct the other individuals by means of this dance language. And again, we have environmental and inherited components. There are some bees that are, due to heredity, more likely to be scouts, and then also the environment influences how many bees will be scouting at any one time. If a colony is really short on food, they will send more scouts out. If the colony has a lot of food, there will be fewer scouts. Okay, with that as an introduction, I want to cover 3 principles of sociogenomics tonight. First of all, that the relationship between genes and social behavior is very close, and that we see evidence of hereditary and environmental influences on gene expression and therefore on social behavior. Secondly, that this relationship, like in evo-devo, is very evolutionarily conserved from animals to humans. And thirdly, something that I call neurons are for today, genes are for tomorrow, the idea of trying to understand how changes in gene expression work their way through those levels of biological organization to cause changes in brain function and behavior. We're only at the very beginning of understanding that third point. Okay, so let's go into the first part, and I want to give you a taste, a highlight of a couple of results, some that are fairly published several years ago now. And the first one is this table right here. This was a case where the work was done by former postdoc Charlie Whitfield. We compared bees that forage and bees that work in the hive taking care of the babies as nurse bees, schematized here. We found thousands of genes that are differentially active in the brain when we compared foragers and nurses, so that if we made a heat map of brain gene activity, there would be lots of genes that would group in one context, nurses compared to foragers. 40% of all the genes that we measured in that first experiment were different, the only difference being the behavioral state of those bees. The differences were so strong and so consistent from individual to individual that a computer program given just the brain gene expression profiles could predict whether the bee was a forager or a nurse without telling the computer which one it was, with almost 100% accuracy, with 1,000 genes in our test case, 100 genes, 50, even down to just 10 genes. The computer could predict out of 60 profiles, 57 or so different accurate predictions. So that was actually our first foray into this entire field. The next experiment that I want to tell you quickly about is a cross-fostering experiment. We cross-fostered bees from a very aggressive strain, African honeybees, AHB, to less aggressive bees that are derived originally from Europe, European honeybees, EHB. We took adults, one-day-old adults, so the basic insect lifestyle, egg, larva, pupa, then they're sort of born or eclose as adults. We took one-day-old adults, cross-fostered them. You can't do that with humans, by the way. Cross-fostered them, and then after 1 week, measured brain gene activity and also looked at other bees cross-fostered in the same conditions at their behavior. And what we found was, is there a heritable component to defensiveness? Yes. Regardless of whether you were cross-fostered in the less aggressive or more aggressive, there were brain gene expression patterns that were due to your individuality. So that's the role of genotype, But there also were genes whose expression varied depending on what colony you were in. Colony genotype, that's environment. So you will recognize, hopefully, this as nature, nurture, both acting on the genome. We saw this for guard bees, that first group of responders. We saw this for soldiers. These are the numbers of genes that are differentially expressed. And we saw this also for foragers, but fewer genes, and that was for the individual genotype, and that was a very pleasing result because African and European honeybees differ in a big way with respect to their aggressiveness, how aggressive they'll defend the colony, but they don't differ as much in their foraging. So we're starting to see that the gene expression really is tracking the behavior in a variety of ways, giving rise to this concept of close relationship. When we expose bees in the laboratory, so a companion experiment, these were experiments, by the way, done by Cedric Alou, former postdoc in the lab, now has a laboratory in France in the INRA system. When we expose bees in the laboratory to alarm pheromone, just alarm pheromone without any other context, it also provoked a fairly large difference in brain gene expression. So we see a close predictive relationship. We see environmental influences. The alarm pheromone results really affect brain gene expression in ways that tracked the differences between aggressive and less aggressive bees, some of the same genes. So we see this same sort of thing that Yehuda saw, where we transpose DNA to RNA. We see the same thing in our aggression studies. And as I said, cross-fostering revealed that there was an effect of the rearing environment, the adult environment, on brain gene expression, with getting technical now, 25% of the variance in brain gene expression due to what colony you were in. And we don't yet know how that works. Is it in the air? Is it in the water? Are they reading political slogans? Like, why do they change when they're in this different society? What's going on there? We don't know that, but we see a robust effect. The final example of a close relationship comes from the foraging side of things, and it has to do with the dance language. So the dance language is one of the marvels of animal behavior. It encodes symbolically distance, direction, and value during those movements that I just showed you. Those waggling motions encode distance, direction, and the value. So bees are able to communicate symbolically with this information and direct others to a food source that those other individuals have never been present directly for. So they rely upon this information. Experiments with robots have revealed that the communication system is about 50% accurate. And being married for over 35 years, 50% communication accuracy is pretty good, I would say. So the bees are doing it with a brain the size of a grass seed. So to probe this idea of how close is the relationship between brain gene expression and behavior, we wanted to see whether the changes in gene expression track components of the dance. We chose distance because the laboratory of Manyan Srinivasan, University of Queensland, developed a really great assay to explore distance perception of bees. Turns out that, and these were experiments that were done in his lab that showed this, bees are able to measure distance by optic flow. You see the checkerboard pattern here? So this is a tunnel that bees were trained to fly through. And what Srinivasan showed was that if this checkerboard pattern was very dense, small checks there, much visual information, the bee would fly through it and perceive that it had flown a long distance. Because the way bees measure distance is optic flow. How much stuff they pass or passes by their retina when they are flying. So they are glued to that information. They cue it into that. So that allowed us to trick the bees. We were able to train bees to fly through this and vary the walls on the tunnel. So a sparse pattern would reflect for bees that it was a short distance, and in fact it really was a short distance. A dense pattern would trick them, make them think they flew a longer distance. This allowed us to ask whether there are differences in gene expression that are sensitive to distance without the confound of the exertion effect. If we did the experiment out in the open and we trained bees to a short distance compared to bees to a long distance, we wouldn't know whether differences, if we found them, were due to perception of distance or exercise effects, exertion. But this experiment allowed this to happen. Former postdoc Moshumi Sen Sarma did this work, and we trained them in this way. Sparse pattern here compared to a dense pattern, the appropriate controls. Were there differences in brain gene expression between bees that thought they flew a long distance compared to those that flew a short distance? The answer is yes. I won't show you the raw data, but here is a treatment of the data where we analyze principal components to find out broad patterns. And what you can see is that, yes, there was a difference. We measured two brain regions, the optic lobe, since this is a visual task, and another region of the brain that you'll be hearing about more continuously tonight, and that is the mushroom bodies, which is a higher order region of the brain involved in multimodal sensory integration. And thanks to some experiments that I'll talk about later, we now know is involved in responsiveness to social stimuli. So you can see, I circled for you, that the gene expression profiles related to bees flying a short distance and compared to the long distance, each of those points represents a brain region, mushroom body, and optic lobe. You can see good separation there. Here are a couple of genes for those of you that are interested, genes that are known to be involved in learning and memory, the Dunce gene, a GABA-A receptor. So we're seeing genes involved in learning and memory, making sense because when bees are exposed, when the bees are measuring the distance, they need to at least remember it long enough to get back to the hive to tell their sisters about. So we see at least evidence for short-term memory. We would reason that there would be short-term memory, and the genes that we see are, in fact, that case. And then finally, wrapping up this first part of the talk, the gentle bees of Puerto Rico. So a former graduate student of mine, Tura Garay, now on the faculty at University of Puerto Rico, made a startling discovery, excuse me, in Puerto Rico a number of years ago, less than 10 years ago. So Africanized bees came from Africa, were introduced to the New World purposefully, but for a breeding experiment that went awry, and then they spread throughout the New World. We know about this because they're very aggressive, and they caused a lot of problems in some of their early spread. But a funny thing happened in Puerto Rico. They arrived in Puerto Rico, they were aggressive, they caused problems, but soon after arriving, they evolved gentleness. They evolved a much less aggressive phenotype. So teaming up with Turu's former graduate student, who was a postdoc in my lab, Ariane Avalos, we did a genetic analysis of these bees, now looking at DNA variation, but I will circle back in comments to RNA in a moment. And a very interesting result was obtained. So if you look at the top, you see how we map something. This is called a genome-wide association study, where you look for DNA variants between individuals that are correlated with variation in behavior. So you find some individuals that are more aggressive, some that are less aggressive, you sequence their genomes, and you do a giant correlation analysis. Because you're doing literally millions of statistical analyses on each and every one of the variants that exist in the genomes from one individual to the other, you have to do some statistical controls to avoid false positives. This dotted line represents the point below which you won't believe that anything is real and above which you take as a significant association. If you pay attention, you will see that there's nothing above that dotted line. When we compared the individual phenotype, whether the bee was aggressive or not aggressive, with the variants in its genome, we found no associations. That's strange, since we know that African bees are more aggressive than European bees. In other words, there are hereditary differences. Why wouldn't we see them in this population of gentle Africanized honeybees? It's not because of a lack of variation, because we did a population genomics study before that, and we found, as I said, millions of variants. So we tried something else. Ariane replaced the individual phenotype with the colony phenotype. You can measure the defensiveness of a whole colony. So beekeepers, and I know we have a few in the audience, and bee scientists will say, this colony is more aggressive than this colony. Not that bee and that bee, but this colony. Colonies have a phenotype. Colonies are a real level of biological organization. They're not just a collection of bees. When we did that, here's the line now. Look at everything above the line. So this is now a classic Manhattan plot. We earned our Manhattan plot. It looks really like that, whereas in our little college town of Champaign-Urbana, we called this the Champaign-Urbana plot. So we graduated here, and the point is that the social phenotype is what's connected to the DNA variants, suggesting an environmental component, suggesting gene expression. And I'm not going to talk about the right panel tonight, but now we are in the process of sequencing RNA and looking at differences in gene expression to see how they match with these patterns of variation. Okay, let's move on to the second part of the talk to talk about the conserved relationship from animals to humans. This work was taking place in the context of the institute that I'm privileged to lead, the Carl Woese Institute for Genomic Biology, where we practice a form of science known as team science. My lab is involved in a gene networks and neural and developmental plasticity thematic research group. We work together on a variety of projects, and this project is a collaborative with these individuals here. Alison Bell studies stickleback fish, Lisa Stubbs, mouse, and then two brilliant computational and statistical genomicists, Saurabh Sinha, computer sciences, and Dave Zhao in statistics. We asked the question that you see posed right here, and that is, do brain responses to social information involve evolutionarily conserved mechanisms? Evolutionarily conserved in the toolkit idea, because the three species are very much separate from each other evolutionarily, and we assume with pretty strong support that their last common ancestor did not have much of a social life. I'll repeat that again later on when we move to the human work. So here are the pretty pictures of the three species: mouse, stickleback fish, and honeybees. The idea is expose the animals to salient social cues, let them respond, let the brain respond, process that information, measure that response by means of behavior, and then measure changes in gene expression. These were the behavioral paradigms, largely around territorial intrusion. We studied both social challenge and social opportunity. I'll be talking mostly about social challenge. So you see the ways that they are instantiated in nature. I'm showing you pretty pictures, but in fact we used laboratory assays. So these are the two laboratory assays that we used. The honeybee work was performed by former postdoc in the lab, Chagai Spiegler, who is Professor Guy Block's PhD student. Bee world, we like to keep it in the family, you know. And so this work is about him, and Chagai is right here tonight, as is Guy, of course. So the idea then is to expose the animals, as I said, collect the relevant brain region, invertebrates, parts of the brain that are implicated in social responsiveness. There's a social decision-making network that was elucidated by Hans Hoffman and Laura McDonald. And so brain regions were chosen based on that. And then in the honeybee, we reasoned that mushroom body would be the right place to look. And as Hagai's work shows, that's exactly the case. So let me just take you verbally quickly through the general results and then go to some data. Are there brain gene expression responses when an animal is exposed to a social challenge? Yes. For honeybee, yes. For stickleback fish, yes. For mice. Do they involve some of the same genes? The answer is yes. And let me just step back and say what a gift it is to be able to answer something so directly like that. What do I mean? Well, in neuroscience, we're plagued by a puzzle. And that is that even though at the level of the cell, there looks to be a lot of conservation, that is a neuron, Drosophila, fruit fly neuron, and a mouse neuron share many, many features. At the level of the whole brain, it's very difficult to articulate regions of those two brains that are similar. We have no clue for how to evolutionarily understand them. That is one an evolutionary cousin of the other? We have no clue. And even making loose analogies is controversial. The brains look very different, and their architecture is different. And so one cannot do this kind of work that I'm talking about using traditional neuroscience. We don't know how many times the brain evolved, and we just don't have that kind of evolutionary ability. In genomics, we can do it. There are statistical algorithms to ask whether a gene sequence in one species is so similar to a gene sequence in another species that when you perform the requisite analysis, you can say with statistical probabilistic assurance that yes, this is a cousin of this one, the technical term some of you will know as ortholog. These are orthologs. Then you can really get going on this. You can look at the gene list in one species, the gene list on the other, say, oh yeah, that one's an ortholog of this one, that one's an ortholog of this one we don't have a clue because we know still very little about genomes, so don't let me oversell it. But for the ones that we do know, we can make those kinds of statements. So, are there genes that respond to social challenge in the three species? Yes. Are some of them common to the point where they are statistically enriched relative to the whole set? Yes. So there is a common genomic, transcriptomic response. What do some of those look like? On the left, some very detailed information about algorithms that were developed by Saurabh and Dave to be able to rigorously analyze this. I won't be talking about those. It's just up here to remind me to tell you that we've done this in a rigorous way, but I'll be happy to talk with any of you offline about this. But here are some of the biological processes that are implicated, various sorts of signaling pathways, heat shock proteins, structural changes that we infer are taking place as a result to the social challenge because genes associated with these biological processes were overrepresented on the lists of common genes. A particular list of interest are the transcription factors. Transcription factors, as I said before, orchestrate the activity of other genes, were overrepresented. There were more of them than what you might expect by chance on the list of genes responding in a common way to a social challenge. So we see this conservation. Can we take this deeper conservation concept to the level of humans, which of course was the goal of E.O. Wilson's book, Sociobiology, published in 1975. It is the task of comparative sociobiology to trace these and other human qualities as closely as possible back through time. Back then, it was not possible to do this. We had some of the tools of traditional neuroscience. As I said, they're not up to the task. So it was beyond science's ability back then to test this. The proposition, therefore, was quite controversial, and it was difficult to get traction. But now, thanks to genomics, we can ask these questions in a very straightforward way, in the way that I talked about for mouse, fish, and honeybee. So this work played out in the following way. Hagai did the social challenge experiments in one summer, did the genomics and so on and so forth, the results that you saw. The next summer, he did the social opportunity results. When he did the social challenge results, he noticed that there were some bees that read the textbook, they did what they were supposed to do, they responded to the challenge. Those are the bees that we analyzed. He also noticed that there were some bees that were not responding to that. Anyone who studies behavior would know, yep, there's variation in behavior, no big deal, let's move on. The next summer, he studied the social challenge, the social opportunity, and for bees, this was the opportunity to rear a larva as a queen. Worker honeybees do not reproduce. All these experiments are with workers. So for worker bees, the opportunity to rear a sister as a queen is as good as it gets. So that's a real social opportunity. It's similar to parental care or even mating opportunities in other species that engage in direct mating, direct reproduction. So a guy collected the bees. Again, some read the textbook. They did exactly what they were supposed to do, and they responded to the social opportunity. But again, some were not responding. At that point, that caught Hagai's attention. We started talking about this, and the idea was, the question was, might there be socially unresponsive bees? Might that be a thing? Might that be a group of individuals? Many alternative possibilities. They could have been on a bad day. They could have had some illness, other sorts of things. Maybe it was just a one-off non-response. So Hagai redesigned the assay to be able to test the same individuals on both assays, not at the same time like I'm showing you with this cartoon, but to give them an opportunity to respond multiple times to the same either challenge or opportunity, and to both. And when he did that, this is the ethogram here that measured that. The answer was yes, they are a thing. So between 10 and 15 or so % of the bees were not responsive to social challenge or a social opportunity when given a couple of opportunities to show what they've got. That's how they responded. This was the full ethogram. I'll highlight two other groups. The group in red are those that responded preferentially to the challenge and not the opportunity. That makes sense given division of labor. There are guard bees, as I've told you before. And then blue, the royal blue, there were bees that responded to the opportunity, but not the challenge. These are so-called nurse bees. So we see clear differentiation. But again, laboratory assay on behavior, other explanations are possible. So in order to move this one step further, we asked whether they could be distinguished by differences in brain gene activity. So on the right, you see, again, a treatment of the data. Were there differences in brain gene activity between guards, nurses, and these socially unresponsive bees? The answer is yes. About 1,000 genes were differentially active. And when you look at those patterns, you can see that the blue cluster with the blue, these are individuals, and the green and the red, more or less a separation. Different symbols represent bees from different colonies. So brain gene expression also showing that they are, or at least suggesting that they are a thing. They're a distinct group. Another line of evidence comes from our barcoding project, where here are what the bees look like when they're barcoded. This is the system, a camera that takes an image back then every second. Now Tim has it up to every two images every second. And under red light, bees and insects can't see red light. And so you see then the study. These are two bees engaged in a particular behavior called trophallaxis. We heard some about this today from several of the talks in that really great student symposium that was referred to in the introduction. This is a social glue in an insect society. Communication, sharing of food, and signaling molecules in the fluid, so forming a social network. So you can use this method to build a Facebook for bees, which used to be a more popular sentence than it is right now, but just to kind of communicate what's going on. And when you build a social network for bees, so this is the glass wall observation hive. Here is a cartoon depiction of a social network. This is not a physical social network. This is just showing connectivity of individuals on the basis of how many times they're interacting and engaging in trophallaxis over a 1 week period of time. So here we have the queen having the most interaction partners, most friends, and the most number of interactions. And that we expect that. So the system is working. The worker bees are attracted to the queen. But I want to call your attention to two different worker bees in the colony, one having almost as many interactions as the queen did, and then another one having a fraction of those interactions. So it looks like this kind of bee would fit the bill for being socially unresponsive. So it's just an inference, but now we've gone a step further. This is the PhD work of Adam Hamilton, a neuroscience graduate student in the lab, used the same method and behaviorally categorized bees from taking from the social network to be able to connect up their occupation in the hive, and their position in the social network. And I won't go through all of them, but you see names that hopefully are familiar to you by now. But the non-responders, that is, put through the assay that Haggai developed, when you look at them in the social network, they have the fewest number of interactions. So now we have direct information that they are interacting in the colony in a way consistent with them being less socially responsive. We're calling them socially unresponsive. So we have confidence, building confidence, that there is such a thing as a socially unresponsive bee. That enabled us then to turn our attention to a comparative question, which is, are the genes that distinguish the socially unresponsive genes, do they have anything in common with what distinguishes humans on the autism spectrum? So we compared, in other words, the gene list from our experiment with gene lists generated from the autism literature. There are two kinds of gene lists. One are DNA, so that is DNA polymorphisms, variants, and then RNA, in this case coming from autopsies, because you can't measure gene expression in live humans in the brain. So what you see in yellow are the sets of genes that show significant overlap between the human list and the bee list. And it was pleasing because we only saw the significant overlap in the genes in the autism gene sets that had the highest degree of confidence in that set, tier 1 through tier 3. This comes from, by the way, from a database created by the Simons Foundation, which funds extensive research on autism. And then likewise, significant overlap between the bee list and the autism list for RNA for the post-mortem cell-based expression. We tried to do some controls to see if this is specific, and so far we only have a specific association for the comparison I made. That is, we take our B gene set and compare with other mental illness gene sets. We do not see the overlap. Conversely, we'll take the autism gene set that I showed you and compare with other B gene lists for other behaviors, and we don't see the overlap. So far, we see it as specific. So let's be clear about what we're talking about here. I've made this point before, but it's especially important to make the point now. I'm not trying to be glib about autism spectrum disorder. It's a very serious and complex disorder. It involves multiple phenotypes, not just social unresponsiveness. So we're not saying that bees are little humans, humans are big bees, nothing like that at all. We're talking about this toolkit concept. And so I mentioned this before, but let me show you now with images. Here we have a tree of life, the basic tree of life that shows evolutionary relationships among the different lineages of organisms, known technically as a phylogeny. As best as we can understand, the lineages that diverged and gave rise to the social insects, ants, bees, termites, wasps, and so forth, and the lineage that gave rise to primates and humans diverged some 600 million years ago, right here at the base of the tree. It was thought to be, the last common ancestor was thought to be a marine flatworm that had a very simple nervous system, not even yet a centralized brain. No social life, as far as we know, then allowing us to then give rise to this articulation of the conclusion, which is that they didn't inherit these sorts of things. They evolved social life independently. And so this is then the toolkit concept that for whatever set of reasons, and we don't know yet, we're still in the stamp collecting phase, the descriptive phase, for whatever set of reasons, nature appears to use and reuse certain molecular pathways. I mentioned them very briefly, but a few molecular pathways over and over again, independently, to create a social brain. And I think a rich area of possible future inquiry is to ask why some pathways and not others. What is it about a social brain that needs to happen that calls upon these pathways over and over again? Okay, final part of the talk has to do with trying to do two things. One is to ask whether these changes in gene expression that we see, whether they have causal implications. So we've talked about changes in gene expression and changes in behavior. And so I've been loose to kind of suggest that that's what's happening. But we haven't proven anything yet. That's called a correlation. We see those changes. Can we be more precise and see how changes in gene expression are affecting behavior? And of course, in doing that, we have to be able to then start to run that through other levels of organization in the brain, because ultimately it's neurons that are orchestrating activity at the neural level, connecting with muscles to actually cause the behavior. So we're developing this concept about how the past predicts the future and the role of gene expression in this. So I'll just start with a very simple picture here. And this shows what happens when you expose alarm pheromone to bees repeatedly every 30 minutes. You see that for the first 3 exposures, so that would be 90 minutes, every time you present the alarm pheromone, you see an increase in the colony's response. Again, beekeepers would say, duh, if you disturb a colony, then you stay away from it for a while because it's in an aroused state. So we nerd scientists proved what beekeepers have known for millennia. But we did it to be able to go on a little further. So part of the changes that occur in the social responsiveness data that I showed you include changes in several different pathways. I showed you some of the signaling pathways. Here's the schematic here. I won't go through the details. I'm highlighting one part. There were changes in gene expression that were associated with changes in brain metabolism. And so we asked whether this scenario that you see here might be true. Social challenge causes changes in brain metabolism. We showed this. Do changes in brain metabolism causally affect changes in the response? So I showed you a change in response, an increase in responsiveness with repeated exposure. Is the change that we see in this particular part of the larger set of changes causal or not? And the answer is yes. Here we have data from honeybees on the left and fruit flies on the right, data that were collected by 2 former postdocs. Claire Richoff now has her own lab at University of Kentucky, and Hongmei Lee, who now has her lab at Lee University in Ohio. And in honeybees, we use the same assay that I showed you before, and pharmacologically injected agents that change brain metabolism in a very predictable way. For those of you interested in the details, changes in oxidative phosphorylation in a way predicted by the changes in gene expression. The dotted line shows the baseline, and you can see a dose-dependent increase in aggression when we changed metabolism in the brain in a way predicted by the changes in gene expression. We get a drop-off at the higher dose, so those of you in physiology recognize a classic U-shape curve. In fruit flies, this was done genetically, so creating a genetic line that using RNAi and specific promoters downregulates the expression of a particular gene involved in brain metabolism, expressed either only in neurons or another set of cells in the brain of increasing prominence called glia. And so that's the bar here. These 2 bars, the controls, represent the parental lines that were used to make the fly experimental strain. When you manipulate brain metabolism in flies, again, in the way predicted, you see an increase in aggression. So this result was very pleasing. The genetic manipulation is more precise. And furthermore, from the evolutionary perspective, flies and bees deploy aggression for entirely different purposes. Honeybees to defend their hive, and fruit flies to compete with each other for food or mating resources. So to see the same brain system engaged, and by the way, a parallel in stickleback fish and mice, it gives us that sort of perspective. When Hagai did the analyses of social challenge and social opportunity, he found that there were some genes that respond to both. So those might be sort of general social responsiveness genes involved in mediating that process more generally, but also some genes differentially responsive. some to social challenge and not opportunity, and vice versa. These are some of the biological processes associated with these differences. You don't have to memorize these. I just want to show you that there's a lot of biology around the fact that there is a differentiation of positive and negative valence. And this then motivated a project performed by Ian Triniello in the laboratory to be able to get at this question of there are changes. We now saw the changes in behavior. So changes in gene expression do somehow make their way through the brain to cause changes in behavior. How does it work? So Ian is looking at valence, the idea that animals respond differently to positive stimuli or negative stimuli, approach, avoidance, all of different sort of thing. One of the fundamental aspects of organization of behavior, and especially important with social behavior. And asked whether, okay, we see gene expressions in bulk samples of taking this whole brain region, the mushroom body, but can we understand this in a little more detail? Are there cells that just express those genes, negative valence genes or positive valence genes, or are there cells that are somehow responding to both? How is it might be organized at the level of neurons? So what he did was focus in on the mushroom bodies for bees exposed to a challenge or an opportunity. I've told you the mushroom body we know is responsive. The mushroom body has been charted fairly well by classical neuroanatomy standards. There are different cells that have their cell bodies inside the mushroom body. They're called intrinsic cells. These are the Kenyan cells, so-called Kenyan cells. There are two categories, KC1 and KC2, Kenyan cell 1 and 2. He used a technique that look at the activity of some genes that are the first genes to respond molecularly, so-called immediate early genes, and asked whether the same cells respond to both challenge and opportunity, or whether there are different cells. By the way, some of the finest work in this area of valence and immediate early genes performed right here at Hebrew University by Ami Sitri using mice. And so we really borrowed some of his approaches for this particular work. So using this method of in situ hybridization, Ian was able to visualize cells that are responsive to social challenge or social opportunity, and indeed found a differential response on the part of two cells, KC1 and KC2. So here you see the data for the first immediate early gene, HR38. Both of these genes came from the social responsiveness, social challenge, and social opportunity gene lists that I mentioned before. And you can see that the KC1 cells respond more to the social challenge relative to social opportunity, whereas the KC2 cells are responding more to the social opportunity relative to the challenge. So that's one gene marker revealing this. The second gene marker showed the same thing. KC1 cells responding more to the social challenge using this marker, EGR, and then the Kenyan cell 2, KC2 cells responding in this way. So now we've identified cells that would have the right kind of profile to fit with the differential gene expression. And now we need a method to bring those two together. And the good news is, bad news is it's just starting. The good news is it's just starting. So we have new methods, some really great methods in neuroscience, spatial transcriptomics, that promise to help integrate molecular and traditional neurobiological levels or neural levels of control. So here's some of our first data using, some of you will recognize I've been talking about single cell transcriptomics. So here is the result of a small experiment looking at 30 minutes post-challenge and 60 minutes post-challenge, and looking at brain gene expression. And some of these clusters of cell, this is now measuring brain gene expression cell by cell for several thousand cells. Some of these clusters have the markers of gene expression that make them very similar to the social challenge response or the social opportunity response, meaning that there seems to be different cell populations that are tracking that valence. Now, we don't yet know whether these populations that look in the way that they should to be Kenyan cells really are, because this first method just takes the mushroom body and dissociates the cells, and we don't know who they are anymore. We just know that they're individual profiles. But the newer methods that we're just getting started promise to bring those two together. And so that will, I think, really help us to get this fuller picture. So when we're decoding the close relationship, I just want to say a couple of words about how might it work. So we see this close relationship. It's connecting genes to behavior in a very meaningful way. So how does this gene regulation occur? So I want to tell you just very briefly about a tale of two transcription factors that begin to give us a picture for how this works. Let me just introduce the subject, though, by telling you or reminding you that one very strong way that genes are regulated, their activity, is that just before the part of the gene that encodes for the protein, that part's called the promoter, in the promoter there are places where transcription factors bind, and when they bind, they orchestrate gene expression. And so one can learn a lot about gene regulation by studying the promoter region and the particular genes or transcription factors that are binding and how they work together. We know that all genes are regulated by the activity of many transcription factors. There is no simple relationship there. So we did a very large-scale experiment over almost 1,000 individual brain gene expression profiles over the course of several years, 48 different experiments in 3 different categories: foraging, aggression, and behavioral maturation. Studied the brain gene expression patterns in collaboration with systems biology faculty member Nathan Price, a student in Nathan's lab, Sriram Chandrasekharan, who now has his own lab at University of Michigan, working with a former student in my lab, Seth Ament, who has his lab at University of Maryland. They created a brain transcriptional regulatory network to help elucidate the relationships between transcription factors and downstream genes and how they connect to behavior. So another one of these so-called hairballs, this time not for trophallaxis, social behavior, but relationships, connectedness between genes. And what was found was there are 2 transcription factors that I want to highlight as one of the results. So what we found was that transcription factor called BROAD was predicted to regulate expression of genes associated with aggression and maturation. Meanwhile, another transcription factor, FTZF1, was predicted to regulate genes associated just with maturation. Maturation means growing up from working in the hive to becoming a forager. But what made this really interesting is that BROAD is predicted to regulate FTZF1. And in fact, not just predicted, we know from Drosophila genetic experiments that FTZF1 is below BROAD in a hierarchy. So how could this be? If FTZF1 takes its orders from BROAD and BROAD is regulating aggression and maturation, how could it be that FTZF1 is just regulating maturation? One possibility is this is all bioinformatics. It's a prediction. It could be wrong. And it could be a more simpler relationship. So Adam Hamilton, former graduate student, set out to test this using RNAi, a method to knock down the expression of the genes in the brain. And he knocked down the expression of BROAD and FTZF1 to test these predictions, bioinformatic predictions. And to cut to the chase, the bioinformatic predictions are accurate. So BROAD was predicted only to affect aggression, and when you use the challenge assay that I've been talking about, you see that when you knock down the expression of BROAD, you decrease aggression in a significant way. But when you knock down the expression of FTZF1, no effect on aggression. NS not significant. Control versus the RNAi treated. By contrast, looking at behavioral maturation measured as the age at onset of foraging, measured, the foraging activity measured not using barcodes, but this time with RFID tags at the entrance, you can see that both transcription factor manipulations cause an early onset of foraging relative to the control, which is green fluorescent protein injections only. So the prediction is there. At the molecular level, we see the same thing. This is a complicated slide, but the message is really very simple. You see these different lines have a different slope to them. Each of those lines represents those three categories: foraging, maturation, and aggression. And the takeaway message is that the relationship between the transcription factor, in this case broad, and one of its target genes is different when it comes from the brain of either a forager or an aggressive bee, and so forth. If it was the same, the lines would be in the same direction. So we're getting then a glimpse of how this complexity is regulated. There's context dependence to the networks, and somehow parts of the network are involved in regulating some behaviors and not others. How that works, we don't know yet, but it gives us at least a glimpse of the picture. More generally, this close relationship between gene expression and behavior perhaps can be seen in this context. Prior to genomics, we kind of had a sense that gene expression and its relationship to neuronal function and behavior was more like a static model. RNA is produced, then the protein's available for neural activity to use it, sort of like a line of taxis that are there. But it looks more like the Uber model, a dynamic model, an on-call, on-demand sort of model, and thereby giving this close relationship. Why the relationship needs to be this close, I think has to do with the neurons are for today, genes are for tomorrow. The idea that an animal takes in a stimulus, it responds to that stimulus, and that provokes two things: the behavioral response and a change in molecular programming that is appropriate for the nature of the stimulus. For a challenge, a threat, ecology predicts that one threat usually follows another threat. So it pays for an animal to become more vigilant. It pays for an animal, if it's exposed to alarm pheromone, case of a honeybee, to become more aggressive and be more vigilant. So we have this, the neurons are there, they respond immediately because they're set by the molecular program. But then the environment gets under the skin, provokes an immediate response for which the neurons are wired to do, but that also provokes a change in gene expression that will cause a change in state. And of course, to continue that analogy, PTSD, the threat never goes away. It's just always there. And so you have a dysfunction of the connection between outside world and the inside world. And we in fact know that there are epigenetic changes that are associated with PTSD that sort of fix the genes in an on state. So going forward, what do we need to be able to take this line of work further? So I've showed it's close, reciprocal, conserved, and causal. To further extend this paradigm to humans, we certainly need to be able to consider the policy implications of this dynamic genome model. If DNA is not destiny, how do we understand the relationship between genotyping individuals and relating their genome to behavioral phenotypes, sort of a lot of dystopian ideas one can imagine by a strict, if you ignore all of this, what we talked about tonight, you can imagine a lot of distorted understanding of the relationship between genes and behavior. And I said, we've seen this movie before in our periods of history. But with the dynamic genome, we have a new way of looking at it that's more complex, but more accurate. We need better ways of measuring human brain gene expression. New imaging methods, perhaps, brain organoids, the idea of taking cells and making brains in the laboratory, also controversial, but also potentially a really great tool for human sociogenomics. And then we need to better understand individual variation in genomes with individual variation in behavior. And as the example from the gentle Africanized bees suggests, we might need to be measuring the behavioral phenotypes at two levels, at the level of the individual and the level of the group, to be able to best understand this, certainly for certain kinds of behaviors, perhaps even including educational attainment. The most recent large-scale human brain genome genotype to phenotype association study. So that'll be interesting to see. The study that was done with educational attainment looked at hundreds of thousands of individuals, so a massive data set, and collectively found gene variants that explain about 10% of the variance. So if you plotted that with one of those plots and asked whether it's more like a Manhattan plot or a Champaign-Urbana plot, it's more like a Champaign-Urbana plot with hundreds of thousands of samples. Maybe that's because the level of phenotyping isn't appropriate, just speculation. So with that, I want to acknowledge the folks whose work I spoke about, many of them mentioned along the way, collaborators, some of the funding organizations, and I'll be happy to take your questions. Thank you very much.
Thank you, Tina, for a dazzling tour de force that even a medieval historian could understand.
Great.
We have a few minutes for a few questions, and then we have a reception downstairs. So if there are anything urgent, please ask, and then you can ask during the reception. Please.
So my intuition is that whenever we have anything that changes behavior, it has to happen at the level of synapses. Because ultimately, that's when neurons communicate. So A, do you have any story to tell about how these genetic modifications are modifying things at the level of synapse? And B, what would you sort of ascribe to genetic modification when you're talking about the environment changing behavior, as opposed to the standard pathways of synaptic plasticity like LPP and LPD?
Yeah, so the question has to do with getting to the heart of the issue with respect to neuroscience, and that is if there's neuroplasticity, you're going to see it one way or another at the synapse, which is the junction between two neurons where communication between neurons occurs. We have no direct experimental information, but when we look at the lists of genes that are differentially expressed in a variety of our different experiments, many of those genes are associated with learning and memory, some with specific synapse bona fides. So we've not shown anything directly, but the gene lists strongly suggest that some of the changes would be occurring at that level. And I think some of these spatial transcriptomic methods, single cell methods, there's talk of even sub-fractioning cells to be able to look at parts of the cell. So there's even a term, the synapsome, to be able to look at that complement of genes associated with synapses. One can right now do nuclei for the single cell, so not just the whole cell, but nuclei, and I think it won't be long before it's possible to isolate synapses and then look at that. Maybe not with honeybees first, probably the mouse or fruit fly that will happen first because one will need genetic markers to be able to do that, but that's where we have to go to be able to address your really excellent point.
I'm more thinking about this isolated bees, unresponsive ones. So, there is some program from the colony level, like the family level, kind of encouragement program, or there is kind of something. What is the fate of those individuals? Where they end up?
Yeah, so the question is, what's the story with those socially unresponsive bees? What's their role in the colony? What's their fate? And so forth. All I can say is that based on the inference that we see from the trophallaxis, the social network experiment, they exist in colonies, and so then they're persisting, and so they're having a life. We don't know if they live for a longer or shorter period of time. I will also say that the notion that there are individuals that work less in an insect society is not new to this work. And one sees this in ant colonies, and one has seen this in bee colonies, and a name has been given to them in some context, and that is reserves. And so the adaptationist idea, which is an evolutionary perspective that everything you see has to be adaptive, has to have been evolved. That's an extreme perspective in biology. But the adaptationist perspective would be that those individuals are reserves, meaning their function is if a calamity befalls a colony and you lose many of the individuals in the colony, they can rise to the occasion and pick up the slack. I'll tell you one piece of information that supports that from our own work. I mentioned that we have the RFID system. We use that to measure foraging activity of individuals. When we did that, we found that about 15% of the foragers, 15%, perform over 50% of the foraging. Sounds like a real elitist sort of picture. But there's a populist twist to that. When you remove those individuals, some of the other bees that were foraging up their game and now start to forage at the elite level. So they might be reflecting this sort of thing where you have individuals that for whatever set of reasons, either at the colony or individual level, either mechanistically or evolutionarily, are not working to their full capacity, and they are then able, under certain extreme conditions, to pick it up. So it's just speculation. Yes?
You show that the stimuli works in an immediate time point and then a bit later. But then in the overall life of the bee, she needs to forget it because she's not responding anymore. So how do you put that in?
Yeah, that's a great question. So you could see an immediate response and then a gene expression response that's on the order of 30 minutes, 60 minutes, 120 minutes. The behavior can persist. The elevated level can persist for 90 minutes, I showed. So then what happens? We assume there's a decay curve that's proportional to a variety of factors. Continued experience, past experience, individual differences. Those play together in a kind of a very rich stew to be able to look at and determine individual differences in responsiveness. We might expect some individuals to calm down and get back to a baseline. We might expect some individuals to hold on longer. We all know people like that. And that's a variety, due to a variety of reasons. So, depending on the stimulus, the nature of the stimulus, one can imagine different sorts of decay curves. One can imagine different baselines. And how they're affected by the individuals they live with and their past and their early experiences and prenatal, so to speak, effects. All of those things would be involved.
In the division of labor, would it affect the division of labor age difference?
It might. It certainly might. We know that when the going gets tough, the tough go foraging. So stress leads to an early maturation in humans as well as in social insects. And so that kind of continued exposure to stress, and if they hold on to it and they're longer, that they could then manifest as growing up faster.
Thank you. We'll stop here.
Okay.
We can continue on the reception.
Okay.